Good afternoon, everyone. We're just going to give another minute or so for people to join the webinar. We'll be live in just another minute or two. Okay, I think we should get started. Good afternoon, everybody. I'm Eric Nestler. I'm director of the Friedman Brain Institute at the Icahn School of Medicine at Mount Sinai, and it's my great pleasure to welcome you to this latest webinar, Decoding the Brain, How New Brain Computers are Unlocking the Secrets of the Mind. When we talk about brain-computer interface, we're talking about devices that we'll hear about today that are placed either on the surface of the brain or even within the brain during neurosurgery, and that can capture the electrical activity of the brain. So, for example, if a person is paralyzed due to an accident or to stroke and can't move an arm or a leg, they might be able to move a device in front of them just by thinking about it. When I started my field a number of decades ago, this notion was pure science fiction. And over the last decade, it has become reality to an extent that has impressed me tremendously. I'm sure some of you have seen some of the headlines about brain-computer interface research in the lay press. And I'm very proud to say that people at Mount Sinai are at the forefront of this effort. And so today we're going to hear from three experts, Josh Peterson, Ben Rappaport, and Ignacio Saez, whom I'll be introducing just before they give their brief pitches. First, I also want to introduce my co-moderator, Dr. Paul Kenny, who is chair of the Nash Family Department of Neuroscience at Mount Sinai, and also the Ward Coleman Chair in Neuroscience and Pharmacology. Paul, thank you for joining us today. I look forward to managing this program with you. So without further ado, let me call on our first speaker. The way we're going to organize our program today is we're going to call on each of the three speakers to give a brief pitch of their work. Paul and I will follow up with a question or two to each of them. We'll have all three speak. We'll have our own questions to ask, and then somewhere around halfway through the hour, we'll turn over to questions raised by you all in the audience. So I would urge you all to use the Q&A button at the bottom of your Zoom screen. You can enter a question that's private, and Paul and I will be following these questions and asking our panelists in the second half of the program. So with that, let me start by introducing my very close colleague for many years, Dr. Josh Peterson. Josh is the chair of the Department of Neurosurgery at Mount Sinai, and also the Leonard Malis and Corinne and Joseph Graber Professor of Neurosurgery. Josh is also the co-founder and executive director of Mount Sinai BioDesign, and he'll be telling you more about the extraordinary efforts of this highly innovative center. Josh.
Thanks, Eric. Paul, welcome everyone. It's a real pleasure to be here. Neurosurgery is a broad field. People may not be so familiar with what we do, but as complicated as the brain, that's also how complicated our field is. We treat conditions that are as diverse as pediatric neurosurgery and brain problems in geriatric patients, brain tumors, neurocritical care, skull base, pituitary, epilepsy, movement disorders, and so on. The list goes on. As Eric mentioned, as the understanding of the brain's functions has evolved and as technology has evolved, the concept of probing the brain's electrical activity and modifying it and measuring it, that has turned from science fiction, as Eric mentioned, into a distinct reality. We started with programs like deep brain electrodes to measure electrical activity and then give pulsations to the brain to stop an epileptic seizure or possibly to control movement disorders. But as we have developed more technology, the number of electrodes that we can use to measure brain activity has grown. The technology has evolved into a spectrum, which on one end has very precise measurements of up to several hundred of neurons at a time with needle electrodes in the surface of the cortex on one end, and on the other end, very minimally invasive technologies in which 16 electrodes are embedded into an intravascular stent and threaded up into a vein in the center of the head, measuring the neighboring electrical activity. What's most exciting to me, and I will share my screen to share some of this with you, is the development of the layer 7 cortical electrode by the Precision Neuroscience company led by Ben Rappaport. And in this configuration, over 1,000 electrodes are embedded into a flexible wafer that is thinner than a human hair and placed on the surface of the brain. The concept being that we can read the brain's electrical activity, transduce and translate that in a computer, and then use that activity to directly interact with the computer and hopefully in the very near future control prostheses and possibly even feel and sense the position of prostheses. Just a warning, I'm going to show you a picture of the brain here. And what you're looking at now is a craniotomy during the resection of a brain tumor that lies underneath the surface. The craniotomy is about 3 inches in diameter, and you can see 2 of the layer 7 cortical electrodes. These round paddle electrodes represented the state of the art up until several months ago. And these electrodes would be used to measure the difference between the motor cortex, which generates activity, and the sensory cortex, which receives that activity. During an actual operation, you'll see that with my right hand, I'm withdrawing the standard electrode, and now we're seeing 2,000 electrodes, 2,048 electrodes reading the brain's activity. We have now used these electrodes to measure and read the signature of individual fingers on the primary sensory cortex. And although it would take a little bit of time to go through each of these, what you're seeing here is an individual signature of each of the fingers. This is the first time that's ever been done. I'm very interested in this field, and you can see that the neurosurgery department at Mount Sinai are really at, I want to say, near the center of the BCI universe, with Ben Rappaport, who you'll hear from, the scientific director of BioDesign and co-founder of Precision Neuroscience, and Tom Oxley, the inventor and CEO, inventor of the Stentrode, CEO of his company, Synchron. If you look at what's been happening in this space, here's a thing that was presented on CNN. Synchron and Precision are 2 of the most important companies in the world in this space. They both lead in the number of patents that have been filed. Precision recently obtained FDA approval to leave the implants in for 30 days. And they've been doing very well with their activity as a company growing. At Sinai, we are hosting the Brain-Computer Interface Symposium. The first one was in November, and the next one will be this October. And so what I see as my role here is to facilitate the recruitment and support of physician scientists like Tom and Ben, to interface with scientists like Ignacio Saez, and under the direction of Dr. Nestler and Kenny, to create a rich environment that we can support these people developing these programs together. So I will stop there, and if Dr. Nestler or anyone has any questions, happy to address those.
Yeah, that's fantastic, Josh. Let me just follow up with one quick question, because I really want our audience to register, what we're talking about. So you would have a patient who has a condition, maybe stroke, or is paralyzed from an accident, and you would be removing a small part of the skull temporarily to place one of these electrodes on the brain. And then from that, you can actually record the activity from that patient's brain. As you said, use computational approaches to decipher what the brain's electricity is thinking, imagining, doing, and then transmit that to a device. It's fantastical.
Yeah. And, I showed you a large opening. That's because right now we're only putting these in while we're doing surgery for other conditions.
Got it.
But this procedure to put it in only requires a very small incision, a slit-like opening in the bone, and slide it underneath. So it does not require a major operation. And yes, then that is threaded to a transmitter, which everything completely implanted, and then a magnet
Like
on the chest.
like a pacemaker in the chest or something.
Yeah, yeah. And then to operate a computer, which itself can then operate either a device or a prosthesis. And keep in mind that that's just the beginning, because once we have an electrode on the sensory cortex, let's say you add an articulating arm that you might then be operating with the computer through your brain's thoughts. You might then be able to sense and feel the position of that prosthesis. So it'd be much more than what we see now with joystick operated prostheses and computer movements.
Yeah. And for me, when in thinking about brain-computer interface, even 10 years ago, I did not expect us to be at this point where it's reality. So this is truly spectacular, and just wonderful to see the leadership role that Mount Sinai with you and Dr. Rappaport and others are playing. Great. Let me turn over to Paul Kenny, who will introduce our next speaker.
Yeah, we've just heard from Dr. Peterson about some of the cutting edge work from our next speaker, Dr. Benjamin Rappaport. So Dr. Rappaport is co-founder and CSO of Precision Neuroscience, which we just heard about. He's also scientific director of Mount Sinai BioDesign, and he's assistant professor of neurosurgery at the Icahn School of Medicine at Mount Sinai. So over to you, Dr. Rappaport.
Thank you so much. Good afternoon. It's a pleasure to be here today. Dr. Pedersen said it all, so I'll try to add just a few insights from around the technology that we've been developing and how it could potentially make a difference in the lives of people living with conditions like spinal cord injury, certain types of stroke, and neurodegenerative diseases such as ALS. As Dr. Pedersen mentioned, as Dr. Nestler was describing, a brain-computer interface is a system that allows the brain to communicate directly with an external device, such as a computer or a prosthetic limb. And the brain, we've kind of taken this for granted in the rest of the discussion, is an electrical organ. So it processes information electrically. And direct communication with the brain requires recording and decoding the electrical activity of the brain in real time, translating patterns of electrical activity into actions in the world outside the brain. But as Dr. Pedersen showed you, the brain is also a physical landscape with hills and valleys and caverns and crevasses. And our approach to interfacing with the brain has focused on developing a device that is both highly precise and reliable over the long term. And you see it here just after manufacture, one example. And we've particularly emphasized minimizing the risks associated with surgery and with the device itself. So these flexible thin films are incredibly soft and pliable in order to conform gently to kind of caress the brain surface. The technology that we've developed is called the LAIR-7 cortical interface, and it consists of an ultra-thin flexible electrode array, which we implant onto the brain surface. And these arrays, as Dr. Pedersen mentioned, and as you can see here, contain over 1,000 tiny little sensors, each about the size of an individual neuron. And each one of these platinum electrodes can detect brain activity at a very fine scale. And so the flexibility of the arrays allows them to conform to the surface of the brain with all of its hills and valleys without doing any damage whatsoever. The idea behind using a flexible electrode is very simple to explain, but actually quite difficult to implement in practice. And it requires very high-tech manufacturing techniques, the kind really that are used to make microchips. And this is a unique feature that has never been possible before in prior generations of neuroscience or technology. And it really represents a very significant technological progress in the realm of next generation medical technology. But in addition to these technical advancements, we are already testing the technology in clinical pilot studies. Some of the most exciting of these clinical pilot studies have been with our colleagues, including Dr. Pedersen and Dr. Sayas at Mount Sinai. And these studies are focused on people with conditions, as Dr. Pedersen mentioned, that currently require neurosurgical procedures that involve according to the current standard of care, electrical recordings, such as surgery for epilepsy or for the removal of tumors. And remarkably, some of these procedures are performed while the patients are awake, allowing us to interact with the living brain in an almost normal state. And I won't spoil Dr. Saez's surprise, but we have something to say about what's happening at Mount Sinai actually tomorrow. But these studies already have allowed us to map and translate the ultra-detailed electrical patterns generated by the brain as a person thinks, moves, feels, and speaks. And I'm going to try to show you an example of that right here. So what you're seeing on the screen is the electrical activity of the brain. These traveling waves of color are just representations of the electrical activity that washes over the surface of the brain in real time. You can see that little red box on the diagram of the brain represents the patch of brain surface that's covered by the electrical array, and behind those dots you can actually see the brain itself, because these electrode arrays are essentially transparent. And that electrical activity, which although it may seem like it's flickering fast here, actually is slowed down about 50 times. So the actual process of thought happens even faster than what you see on the screen. And these studies have allowed us to begin decoding the process of thought. This type of image that I just showed you until about a year ago had never been seen before in the world. No one had ever seen human thought resolved at such a fine level of spatial detail. And we've shown in some of these studies that we can decode the thoughts and intentions of patients in real time, predicting their intentions to move and to control their own hands, as well as robotic prosthetic hands. And what we learn in these studies is informing how we translate these thoughts and electrical signals into the actions and communications with the digital world, like what Dr. Pedersen and Dr. Nestler were saying, to help patients with paralysis from spinal cord injuries, stroke, and ALS. All of these three conditions are examples of conditions where people have lost some or all of the ability to move or communicate, and brain-computer interfaces can help such patients by reestablishing the connection between the brain and the outside world. The idea of a brain-computer interface may have seemed like science fiction or laboratory science even a couple of years ago, but right now it's a clinical reality at Mount Sinai, and over the coming years, we all expect that it will become a standard of care, helping patients regain lost abilities to return to independent life after major neurologic illness. Thank you all for having me here and for your attention, and of course, I'd be happy to answer any questions.
Yeah, that was wonderful, Dr. Rappaport. It's amazing to see the brain in action. We heard a little bit about the patients involved in your clinical studies, but maybe you could tell us just a wee bit more about the patient populations who are currently enrolled in your studies and those who you think will really benefit from the technology that you've been developing.
Absolutely. So, because the brain is an electrical organ, a lot of the standard of care in clinical neuroscience involves diagnostic and therapeutic procedures that involve electrical techniques, not so different from what many people are perhaps more familiar with in cardiology, where you go for an EKG and you have some kind of electrical test. But seeing the output of that has always been very difficult for the layperson to, even for specialists, even physicians have a hard time reading the electrocardiogram. And in the brain, it's actually even more complicated, right? So what you do with 1,000 electrodes on the surface of the brain without artificial intelligence is, has been almost impossible to decipher. So it's really only in the modern era that we can begin to decode the kinds of signals that we're seeing at such a high resolution. But to answer your question, because the standard of care in neuroscience gives us access, electrical access to the brain so often, we have been in the clinical pilot studies studying patients, for example, with brain tumors or epilepsy or movement disorders like Parkinson's disease. And I would say some of the most fascinating of these procedures, Dr. Pedersen showed you some examples of where we've been able to map, for example, where the fingers are in a patient who has a brain tumor just near the finger area. And that kind of application is incredibly important because you want to make sure you don't damage the area that controls the fingers when trying to remove a tumor from just nearby. And doing that in a patient while they're asleep and not able to move their fingers to show you where they're living in the brain is very, very important. And we've now been able to do that at a resolution that's much higher than was ever possible before. But in some of our awake patients, including one that we're going to see tomorrow and others that we've handled while awake, some of those patients are able to speak and move actually while the surgery is taking place. And that has allowed us to place the electrodes on the motor cortex that controls the area of their hand and face while they're speaking or or intending to move or imagining movement. And in those patients, we've been able to learn what patterns of electrical activity correspond to the intention to move and drive a prosthetic or drive activity in a digital interface based on thought alone. And we've done that in these patients, the Parkinson's patients or the tumor patients or the epilepsy patients, because that's the population that the first generation of clinical studies has given us access to. These are, I want to just make it clear, these are patients who are really, I would say, very brave and altruistic, and they're engaging in these studies in a way to really to benefit patients like themselves in the future, right? They know that the device will be implanted, removed, something will be discovered or validated in the process of their treatment, and it will go to help other patients in the future. And those future patients, not so far into the future, are patients with paralysis who can think but can't actually move their hands or move their speech apparatus to make speech. And those are the patients who will benefit most from a permanent implant that allows us to translate thoughts, the electrical activities that correspond to thoughts, into actions, a speaking avatar, or movement of a mouse in a computer interface, or typing, or thought to text type interfaces.
Yeah, it's just incredible. I'm sure we're going to have many more questions, but I think I'll pass it back over to Dr. Nestler for our next speaker.
Thank you very much. And our third panelist is Dr. Ignacio Saez, who's director of the Human Neurophysiology Laboratory within the Friedman Brain Institute. He's also an associate professor of neuroscience, neurosurgery, and neurology at Mount Sinai. Ignacio, I'll turn things over to you.
Thank you so much, Eric, and pleasure to be here and be speaking to you. So you've heard a little bit about the neurosurgery from Dr. Betterson. You've heard about the neuroengineering from Dr. Rappaport, and today I'm here to tell you about the neuroscience. So as you've heard, neurosurgical interventions provide an amazingly detailed depiction of human brain activity that is not achievable in any other way. We can register electrical activity thousands of times per second in hundreds or even thousands of independent brain locations. And my research is fundamentally focused on understanding what that brain activity means. How these electrical signals generate human thoughts, human feelings, actions. How is the brain operating when you remember, decide, and imagine? And importantly, how are these processes affected in neurological and psychiatric conditions? And of course, these questions are intimately connected, because we won't be able to help paralyzed patients to move unless we understand how the brain generates movement. We won't be able to fully treat depression without understanding how the brain generates move, et cetera, et cetera. So these are questions that have been under intense examination in neuroscience for a long time, but these new methods that we've been discussing today are providing novel insights into human brain function, which will provide knowledge that is just not accessible in traditional animal experimentation. We have derived very fundamental insights into brain function from monkeys, rodents, and other animal models, but of course there's limitations associated with those experiments. There's complex behavior that animals cannot carry out. We cannot know how they feel because they can't talk to us. And there might be differences between species, etc. So by combining neuroscientific methods and questions with these novel neurosurgical techniques, we can have a unique view not only into the human brain, but also into the human mind. And I'm going to give you an example for something that we recently published from our own group at Sinai, in which we had patients with electrodes surgically implanted in their brain for treating their epilepsy, coming to their lab and complete a meditation session. The patients are in a very nice wood paneled room with an experienced meditator guiding them through the session, and at the same time, we're recording what is happening deep in their brain. So we focus on these ancient techniques because they're known to have a substantial positive impact on mental health and can potentially provide better understanding of what good brain states look like and potentially lead to development of future treatments. What we found in this study was that patients could willfully modulate brain activity in the hippocampus, which is a brain area involved in memory, and the amygdala, which is a brain area that is involved in emotional regulation. They also reported feeling better, more calm, and less anxious. And now, for the first time, we have a picture of what happens in the brain while patients transition to this positive emotional states. So we're in the business of turning that data into knowledge about the patient's brain state, but what else can we do with it? Beyond the possibility of kind of self-regulating, as in the meditation study, we can also think about building brain-computer interfaces that directly connect the human mind with a computer, and even potentially an artificial intelligence. We can go even further and leverage the fact that these methods provide not only a passive window into brain activity where we're just listening to what the brain is doing, but also the activity to modulate it through electrical stimulation, a technique that we call neuromodulation. And unlike pharmacological treatments in which we take pills that flood the brain with chemical compounds that globally modulate brain activity, these targeted neuromodulation interventions allow us to specifically target individual brain regions which we know serve specific functions and are differentially affected in different disorders. For example, we can target areas that participate in generating these mood states to treat affective disorders. Like Dr. Rappaport alluded to, tomorrow we're going to carry out for the first time a new experiment in which we're going to use the Layer 7 device with the goal of recording electrical activity from the prefrontal cortex in a patient. And this prefrontal cortex is a brain area that's uniquely developed in humans. So this provides a unique experimental opportunity. And we're going to relate this brain activity that we're going to record to patient actions and mood states. So really what you're seeing today is in real time, the cutting edge of the combination of neurosurgery, neuroengineering, and neuroscience that is happening at Mount Sinai today and tomorrow. And our efforts to develop new brain-computer and stimulation interfaces for treating brain disorders that are in dire need of new treatment options so we can help our patients live healthy, happy, and fulfilling lives.
Mhm. Thank you, Ignacio. I wanted to ask you a question. Roughly how many nerve cells do you need to record from in your studies, from a person, in order to infer something meaningful about their behavior, whether they want to move something or think about something or feeling something? Is it hundreds, thousands, tens of thousands? How many nerve cells do we need to capture to do this?
Well, the answer, of course, the more the better, right? So we've been able to do really well with a small number of electrodes, but the new technologies that Dr. Pedersen and Rappaport introduced are going to fundamentally change the landscape of what we can do, and they're going to tremendously enhance our ability to record. So to some extent, it's kind of difficult to anticipate exactly how much more we're going to learn from these, but imagine that you have access to the kinds of thick electrodes that Dr. Pedersen highlighted in his talk. That's like looking at a few flashlights that turn on and off. And now substitute those flashlights by a whole TV screen where you can see images. You can really get a detailed depiction of what's going on with two-dimensional activation and temporal resolution and so on. That's the kind of game-changing technology that we're developing at Mount Sinai that we think is going to lead to fundamental new insights into how the brain works. It's kind of difficult to know exactly what's going to happen, which is one of the reasons why this is so exciting.
Yeah, no, absolutely. Thank you. Thank you very much. So let's now move to the second part of the program. Paul and I are going to ask our panelists a few questions. I'll start with a question for Dr. Pedersen. Josh, tell us more about the various types of electrodes. If I remember correctly, you talked about actually putting an electrode through somebody's vein, if I gather correctly.
Right.
And that ends up somehow in the brain, recording from the brain. How does that happen?
Yeah. The question you asked to Dr. Saez is a really interesting one. You can get very precise recordings actually from a single neuron with a needle electrode, and these are the sort of electrodes that are used in the Neuralink type of interface that's Elon Musk's company. Of which Dr. Rappaport was a co-founder, by the way, till he moved to Mount Sinai in a very intelligent move that he made. And that's really interesting because you can get very, very precise information from just a few neurons that surround that, very precise. But that's also a little bit invasive because you have to put that into the brain. But what if you didn't want to even open up the head at all? Could you thread an electrode through the blood vessels and into the brain and near the brain and near the brain's electrical activity? And that's what's done with the stentrode. I'm sure that people are familiar with the concept of cardiac stents that open up the coronary arteries after or before to prevent a heart attack. And similarly, we have those now for the carotid arteries and the intracranial arteries. synchron embedded 16 electrodes into a stent that's threaded through a vein, either accessed in the arm, the neck, or the femoral artery, up into the primary vein that drains the brain, the superior sagittal sinus, right in the middle. And as it turns out, if you look at the representation of the arm, the hand, the mouth, and the leg in the motor cortex of the brain, the leg is draped over the cortex right in the midline, right near that vein. And so if that stentrode can be delivered and expanded and permanently implanted so as not to obstruct blood flow, but to position those 16 electrodes near the leg area of the brain, patients with neurodegenerative disorders like ALS, Lou Gehrig's disease, who don't have the ability to transmit the activity, can teach themselves how to move a mouse. And when you talk to them, what they say is, well, when you told me to move the mouse, I imagined that I was going to move my hand, but that didn't work. But when I realized if I kicked my leg, of course they can't move their leg, but they imagine that they're kicking their leg. Now the mouse moves because the leg cortex is near the electrode. It activates the electrode. It's translated into computer activity. And now the mouse moves. And they train themselves so well that they can type with their leg. But of course, they're just intending to move their leg, which then types a word, and they can communicate with caregivers and so on. So it's an amazing thing to watch this happen in real life. But of course, we feel that putting 1,000 or 2,000 electrodes directly on the area that generates those thoughts to move things is going to be far greater.
Right. It really is phenomenal. And I am just imagining the promise and hope this offers patients who are paralyzed due to one of the conditions that Ben and you have mentioned. Paul, go ahead. You ask the next question.
Yes, it's something that Dr. Peterson just brought up, and that's the fact that Dr. Rappaport previously worked at Elon Musk's company. I guess I've got to ask, how does your technology compare to what Elon has been developing?
Well, Dr. Beddison kind of alluded to it, that one of the things that we really tried to engineer beyond was this limitation of needing to implant needle-like electrodes into the brain for each basically increment of bandwidth, increment of information, of increase in the information stream that comes out of the brain. And if you think about the way those penetrating electrodes work, or really if you think about what these brain-computer interfaces are, they're really a communication interface between the brain and the outside world. And as everybody knows nowadays, this concept of bandwidth, which is really information per unit time, is just so much a part of our everyday lives. And we know that that it's only, for example, over a high bandwidth connection that we can even have this webinar, right? This is not something that we could have had over a dial-up, in the 1990s, right? You couldn't even think about that. But if you think about the technology that we want to open up the floodgates of data between the brain and the outside world, to really have natural, fluid communication, and to build function on top of that, channel of data, you need a type of interface that basically can expand almost indefinitely in terms of the data throughput. And so when you have needle-like electrodes, you're kind of limited in at least two ways. One is that every little needle does a tiny little bit of damage to the brain. And so at some point you have to stop implanting needles, because the amount of damage when the number is small is sustainable. But when you get to large numbers of neurons and not large numbers of electrodes, you just can't keep scaling that up. And so you can't grow the bandwidth indefinitely. So at Precision, we really worked very hard to build an interface that didn't do any damage to the brain surface, which means that as you scale up the number of electrodes, basically almost indefinitely, you can just keep increasing the amount of information transmission, the bandwidth between the brain and the outside world. And actually, last year around this time, Dr. Bederson set the world record for the number of electrodes placed on the human brain. At that time, it was 4,096, and I'm sure he's eager to break that record at some point, which we hope to enable. But it gives you the sense that the platform that we have developed at Precision has an intrinsic safety, and that's kind of a little bit proven in the fact that we were the first company, in the modern BCI era to get an FDA clearance for this electrode, and that's allowed us to scale the information transmission. And so, what does that allow us to do? What's already allowed us to do is we've already been able to interface with all different regions of the brain that you couldn't even imagine touching with the Neuralink device or with, for example, the Stentrode. We have been in contact with and integrated with the motor cortex, which controls movement. As Dr. Bederson showed you, we have mapped the sensory cortex, which controls sensation at, basically the highest level of detail that's ever happened, as Dr. Bederson showed you, individual finger level. At other sites, we have, including here at Mount Sinai, we have had the electrode just basically against the brainstem, which is a part of the brain that no one would ever think to touch with a Neuralink electrode. Also, Dr. Bederson has shown in his work with the precision electrode that the precision electrode actually can conform to the hills and valleys of the brain. So not just the most superficial aspects of the cortical surface, but actually those gyri and sulci that make those curved contours that we all have seen in pictures of the brain, and the Neuralink electrode can't do that, but lots of brain surface is tucked away in those valleys. And so the precision technology allows us to safely access all of that. So that's just to give you a sense, of how what we've done has been different. And it's really oriented towards being scalable, being extremely safe, and being forward-looking, so that we can, basically produce a platform technology that gives us access not just for restoration and paralysis, but as Dr. Tsai has mentioned, to even think more broadly towards future applications in other types of neurological disease.
Thank you. Perhaps I can ask another question, Eric, if that's okay.
Yeah, sure, of course.
This is for Dr. Saez. I mean, I'm a basic neuroscientist, so I see the types of experiments that you're talking about all the time where, we record from mice or we record from rats, and we collect those types of data that you were talking about, you were describing, on occasion even from monkeys, but very, very rarely do I ever hear about the types of experiments that you're conducting where you can record from the human brain in living patients and really understand how the brain is working. Maybe you could tell us just about kind of how unique, presumably, that is.
It's an amazing opportunity, right? And at multiple levels. One is that, humans are pretty smart, right? And we're capable of a behavioral repertoire and sophisticated behavior that animals are just not capable of, or it would be extremely difficult to study, right? But perhaps more importantly, when I ask my colleagues, such as you or other folks at Sinai that are working with monkeys, and I said, what can I do that you wish you could? The answer is invariably, I would ask them, right? I would ask them because it's relatively easy, and nothing is easy, but it's comparatively easier to understand something that can be observable, like a movement, right? If a monkey is reaching and grasping and moving their arm, we kind of know exactly what's happening in the external world. But when we're thinking about internal brain states, which are potentially very, very important for us to understand from a pure basic and also a translational perspective, like mood is a great example, right? How do you feel? It's not necessarily observable, but it is so intimately important to a lot of psychiatric conditions, such as anxiety and depression and so on. It's very, very, very difficult to study that in animals, or at least there's significant limitations that we can neatly circumvent by working with human patients who can tell us, they can report. They can say, I'm feeling great, I'm not feeling good, or I'm trying to do this, or here's the strategy that I'm using, right? So we kind of have, in a sense, ground truth to relate our electrical recordings to in a way that it's otherwise not possible. So the combination of those two things, the sophistication of behavior and how close we are to the actual internal state that is otherwise not observable, make these amazing experimental opportunities, which literally cannot be done in any other way.
Thank you.
Yeah, so let me follow up on that point, actually. And I don't know if Ignacio, if you want to answer this or if Ben and Josh want to weigh in. By the way, we've gotten several great questions from the audience, so please keep the questions coming in the Q&A button at the bottom of your Zoom screen. We'll get to as many as we can. A high school sophomore put in a question. It's great to see high school students attending the webinar. High school student is interested in emotional trauma and its impact on the brain, and is wondering whether this technology in the future might even be used to help somebody with hard to treat post-traumatic stress disorder, for example. I know that may be a long way in the future, but can you imagine an era where interpreting what the brain is, how the brain is acting and behaving electrically, can be then fed back to a device to correct the circuit to treat these types of conditions? So Ignacio, you give the first try at that?
And then everybody else can jump in. I think the answer is absolutely, right? So we can think of, one way to think about PTSD is as a very salient but maladaptive memory, right? This really crucial event that trigger these memories that just won't go away. And so we know a fair amount about what those memories look like, and that gives us sufficient clues to go studying exactly what's happening in conditions such as PTSD. But more importantly, PTSD is an acquired condition, right? Something traumatic happened and a healthy brain became a damaged brain in a way that potentially is not irreversible, right? We don't know how to yet, but we can think of these BCI interfaces and these neuromodulations as not something permanent, like a prosthesis that's going to be implanted forever, but maybe something that can help us, some sort of mental crutches that can run that change, which is really a maladaptive influence on synaptic plasticity. So changes that happen in the brain was okay, and now it's not okay. Potentially, we can use these devices to kind of run that in reverse and help the brain de-learn those maladaptive memories in PTSD so that it's reverted to a healthy state. The same way that we know these things happen moving forward, we can imagine them moving backwards with the temporary aid of some of these devices that then maybe by leveraging the intrinsic ability of the brain to modify itself can go back to a healthy state. That's how I imagine it. I don't know if Dr. Rappaport or Betterson have more to add.
And Josh and Ben, if you wanted to expand, a couple of questions are focusing on memory as well, whether devices like this could assist one day in people who need help with their memory.
One thing I'll say is that one of the things that we're very excited about is that the restoration of function in people with various forms of paralysis to us is the clearest near-term target. It's a clear and present need, totally unmet, and it's an area that we already have proven that we can make a major impact in. But on top of that, as we kind of have shown in some of the examples that Dr. Betterson and I showed, the precision technology is really a platform technology that allows exploration and discovery. And so even as we've been engaging in the work that we need to do to make this a product that's part of the standard of care that physicians and surgeons can prescribe and implement to really treat people with various forms of paralysis, we really are working already to enable discovery in other areas. And so I can't say that I know the answers to some of the questions like how is this going to help patients with depression or PTSD or disorders of memory or other sort of higher order cognitive issues. But I'm very excited. I know the Precision team is extremely excited in the potential of the platform to enable discovery in those areas. And we're working with Mount Sinai and other sites across the country to just put the tool in people's hands and see what we can discover.
Yeah, if I could add on to that, one of the really interesting questions that has been asked a few times, and someone asked it here, Monica Starkman, the way that these electrodes are placed actually is going to allow us access to the deep areas of the brain. And what you can see here is that the limbic system, which is relevant in many of the psychiatric disorders, depression, OCD, and others, is located here between the hemispheres. And slipping one of these electrodes down in there, I think actually is going to be very easy and very safe. So in terms of access, we should have access to the mood and anxiety areas. As to memory, we can hypothesize, but it's known that we have to activate the reticular system to remember things. And we don't really know yet if stimulating these electrodes will have an impact on function. But as we explore the sensory portion of this experiment, in which we intend to stimulate the sensory cortex, we will gain experience with activating the brain in addition to recording it. So your question is not answered yet, but we're certainly going to be pursuing it.
Well, maybe I've got a question down, I guess. Not my question. It's actually from one of the participant audience members. It's from Naoko. This one's for you, Ignacio. So what distinguishes pure thought from specific or intentional thought directed towards action items, such as moving one's limbs or speech? We have many thoughts, but we don't verbalize most of them. Can we distinguish?
Yeah, great question. And it's very related to some themes that we've touched on, right? Between observable choices and movements, like limb movements, and more abstract types of thoughts and reasoning, right? So that's a question that I'm deeply interested in, and we have been studying in my lab for some time in the context of economic decisions, right? So imagine that you have two different options, which could be, chocolate or strawberry ice cream, or in the way that we have people play the game. Do you want a safe reward in your pocket, a $10 prize, or do you want to flip a coin for $30, right? And we can have people play these sorts of games, and we can make very sure through experimental design that those choices are not purely motor in nature, right? Like sometimes the risky thing will be on the left, sometimes it will be on the right, and so we can kind of dissociate the purely sensorial and motoric aspects of the task from the abstract reasoning that goes into thinking, I want these or I want that. And the answer is that we can do very well at separating those in a variety of brain areas, which is also another thing that we've been talking about, that some of these processes are not just located in a single brain area, but they're actually distributed. And so by leveraging the neurosurgical interventions and these electrophysiological recordings, we've been able to build machine learning models that actually predict what the patients are going to do before they do it with pretty high accuracy. So we're listening to the brain activity, we're running it through a model, and we take that snapshot about a half a second before the patient decides, I'm going with A or I'm going with B, and we ask this model, what's the patient going to do? And again, it's in a very abstract sense that is not directly correlated with the motoric aspects of the task. The model can tell us, based on the brain activity, what the patient is going to do, which is amazing in my mind, but also it lays the groundwork for defining exactly what a brain state is, right? In this abstract sense, which may be decisions, it might be memories, it might be maladaptive memories like PTSD that we talked about before, or any other sort of pathological or cognitive function. So it definitely can be done.
Excellent, thank you. One of our other audience members who said that they're quadriplegic has this question, and I think Drs. Rappaport and Peterson might address this. I would like to know what I should expect from the first commercialized BCI and how they will impact my life from either a social, entertainment, or work perspective. A really good question. Ben, Josh, who wants to go first?
I could just give you a little taste of that. Thank you so much for that question. We don't make progress without your help. You participating in this process, helping us identify what directions we need to go in, describing your experience if you have this, and helping us improvement have been the fundamental step that has allowed us to progress. What I learned from one of the first patients who had the Neuralink implant, while it worked for the first few days, was that he immediately, and he was a cervical quadriplegic, he wanted to play video games. And the very first thing he did was get on his computer and started to play video games, because he'd not been able to interact with a computer the way that he wanted to. And he was very, very happy with this for the first days. And as the signal started to decrease, we think possibly due to an inflammatory reaction related to the penetration of the cortex, one of the reasons why we're pursuing the surface electrode, that signal started to decrease. And as I understood, it decreased down to about 15%. So I'm not sure if I'm answering your question, but I do think that the ability to interact with the computer in a way that's meaningful enough to play a responsive video game would be pretty substantial. Ben, do you want to comment further?
Yeah, I would love to. I want to echo your thanks to people with lived experience for being a part of the design process and helping the scientists and engineers and physicians really have deeper insight into what is the threshold for an implantable device to meet the needs that you or others like you might need. And we have spent a lot of time trying to understand this and the needs are somewhat consistent in our conversations, but they differ across different types of neurological injury. So for example, people with ALS have needs and desires that are slightly different from people with injury of the cervical spinal cord, have needs and desires that are slightly different from people with brainstem stroke. But in terms of the functions that we're targeting in the first generation system, just to be really clear, we've held ourselves to a goal of trying to enable a level of performance that could, in the near future, enable somebody who held a desk job and lost it because of their injury to go back to the level of functional interaction with a computer interface that would allow someone to control most desktop functions at or better than the level of their non-injured self. And we do think that is achievable in the near future. Actually, we think that we in the field are pretty close to that. And with that comes things like entertainment and some ability to have enhanced levels of independent communication with loved ones and friends and so on. But the kind of, I would say, sanity check benchmark that we've tried to target is restoring the ability to hold a desk job in the way one might have but prior to an injury for many reasons, just because it's in some ways it's easy to explain, but also the social, financial, and implications and the implications for a level of independence and the medical economic justification for treatment at that level just makes sense to so many of the stakeholders, most importantly, the people with lived experience.
Yeah, fantastic, Ben. We have time for just one more question. Dr. Peterson, let me ask you this. This is from the audience. Do you think we need BCI to recover from paralysis, or can neuroplasticity, in theory, suffice? Maybe just give a quicker answer than normal, because we are short of time.
Fantastic question.
Yeah.
I'll bring you back to the first thing we discussed. The person who learned how to operate a mouse with their leg was, in fact, exhibiting neuroplasticity. And because he had to retrain his cortex to do something completely different. So neuroplasticity is going to be absolutely key here. Training part of the cortex next to the fingers to start learning to use that part of the cortex to move the fingers. So it's going to be a combination of the machine and the host working together and teaching each other.
Right. Fantastic. So with that, let me thank our three panelists. Dr. Kenny and I are so appreciative of your participation. Let me close with just a couple of remarks. For those of you who are more curious about neurosurgery, I think most of us who are not neurosurgeons, it's hard to imagine what it's like to being in a neurosurgical operating room and working with an individual, I want to draw your attention to a program that we have coming up in a couple of weeks on Wednesday, May 7th at 6 p.m. This takes place at Mount Sinai West. You see the address on the screen. And this is essentially an open house. Come by our world-class clinical neuroscience center and have leaders in neurosurgery, neurology, psychiatry, and neuroscience tell you more about how this work is actually done. With that, I want to thank all of you for joining us today. The work that you've seen is truly world-changing. You know, I am especially proud of the fact that we here at Mount Sinai are writing scientific history. This is incredible. And this work requires a lot of funding, particularly during these days. Most of you, I'm sure, know that NIH is having particularly difficult times, and the future of the country's biomedical research enterprise, upon which we depend so much for our physical health, mental health, but also our economic health. Your generous philanthropy is more important than ever. So please do consider going to this flow code here and contributing to the efforts of the scientists and doctors whom you heard speak today. With that, thank you so much for joining us. Have a wonderful evening. Bye-bye.
Thank you.